In the last video, we ended with a vesicle being transported to the Golgi body. In this video, we'll pick up from there and talk about what occurs once the vesicle arrives at the Golgi body. The first thing that happens when a vesicle arrives at the cis side of the Golgi body is that the vesicle fuses with the cis cisterna of the Golgi. This fusion occurs in a few key steps. First, a tether protein tethers the transport vesicle to the organelle, in this case the Golgi. Here, we can introduce the snares, which are integral membrane proteins whose primary roles are to mediate vesicle fusion. The two main types of snare proteins are vesicle snares and target snares. After a transport vesicle has been tethered to the organelle, a vesicle snare helps dock the vesicle on the Golgi membrane. Next, a target snare helps the vesicle fuse with the Golgi by first going through hemifusion, then allowing for a full fusion. Once the vesicle fuses with the Golgi, its protein contents can be released into the Golgi apparatus. The Golgi is one of the most important organelles in trafficking because it helps the proteins reach their necessary final destinations. At the Golgi apparatus, additional modifications are made to the transported proteins by resident Golgi enzymes. These modifications are key in ensuring the proteins reach their final destinations once they leave the Golgi apparatus. The Golgi apparatus is divided into several distinct regions. These include the cis cisterna, nearest the ER, and the centrally located medial cisterna, the transcisterna, and the trans-Golgi network, or TGN. According to the cis maturation model, the proteins are transported through the Golgi stack as the cisterna containing them migrate, or mature, in a cis to trans direction. New vesicles from the ER continually supply new cis cisterna as trans-Golgi network vesicles mature. Within the trans-Golgi network, proteins are sorted and grouped by their final destinations. This sorting and grouping is accomplished by receptor molecules embedded in the membrane of the TGN.